All right, so next little set of lab activities that we're going to be doing is kind of really hands-on things. So it's going to be tough for you guys who are completely virtual. I'm going to do my best to kind of show you some of the things that they're going to be doing, because we're going to do some practice loading of gels. So if you had myself or Mr. Blue in first year bio, or maybe did it with Mr. Dilks or Miss Justice, I'm not quite sure what they do on that side, you guys did some sort of gel electrophoresis. It's kind of like chromatography, which means that you are separating out molecules, but here we're separating out proteins, we're separating out DNA. And instead of solubility and molecular mass, we're going to use these gel boxes that we see here on the table. And inside these gel boxes, I'm going to make one for you here in a moment. We're going to put this little white powder. This is called agarose powder. And I mix it together with some buffer to help maintain the pH, so it's kind of like a Gatorade solution. And I boil it. And I get this particular kind of, it's almost like a jello mixture, but since it's still very, very hot, I can mold it. And that's basically what I'm going to do now. So I'm going to go back to using those pie pumps that we did before. I'm going to pump about 10 mLs into our little molds that we're going to make here in a moment. So this gel box lid opens. You guys are going to see a couple things on it. It has actually a little platform in it, and this little platform has little red lines in it, and that's where the wells can be made so that we're able to see them. Now when I talk about wells, it just means openings or holes. So that's going to go in there, and what we're going to end up doing is I have these little dams that I can put on both sides of that platform, and it kind of blocks it up so that I can fill it with that agarose liquid that we have there. It's kind of like the jello. So that goes in there very tight. So now I can kind of go through, hold with my hot hand, because like I said, it's kind of hot that we have. And I'm going to pie pump 10 mLs of my boiling liquid, my agarose liquid. Now agarose is just a sugar compound that we have. And this liquid, once it hardens, is going to become our matrix, meaning what our molecules are going to move through. Just like in our chromatography lab, our chromatography paper was the matrix. Here's actually physically going to be the gel, and the molecules are going to move through the gel, not on the top, not on the bottom. They actually will run through it, and they can move faster or slower depending on how much agarose powder I put in or the concentration that I put in. So we'll kind of fill that up. I'm going to give it just a little bit more. Now I need to make the little openings or the wells within it. So these boxes are great, and they actually come with some little combs, we call them, inside the boxes. And these little combs, when they go into the liquid, will actually, once it hardens, form little molds into it. And I'm going to put them for wherever I want those wells to be made. Do I want to make them at one end? We'll talk about the black end of the box and the charges of it. Do I want them at one end? Do I want them at the red end of the box and the charges on them? I would stick it here. Or am I not quite sure what the charges of the molecules are, in which case I would put it smack dab in the middle and kind of holds it in place. We're doing practice loads, and since I'm making a ton of gels, I'm going to give you guys a ton of wells to practice on. So I'm going to throw 3 combs into my practice gel. That way I'll have a ton of openings for you guys to go through. So this takes about 10 minutes or so to go through and harden. So I'll show you guys that one more or less towards the end. So what they're going to be doing this, hang with me here as I move because we're going to the other table. We are going to practice loading these, but I want to go through and practice some other skills first, because now we're going to go back to those micropipetters that we utilized, and we're going to use a micropipette, and we're going to set it to 8 ml. So notice this is my 2 to 20 micropipetter. I have 8 microliters set on it. I'm going to go through, I'm going to seat a tip onto that micropipette. I can pretty much keep the same tip all the way through here, and I am going to be loading it with what is known as loading dye. So loading dye is just a solution that allows me to see that I'm putting it into the opening or the well easily. It's also denser than the buffer that it's in. So as long as I'm over top of the opening, this is going to sink into the opening or the wells. So we're going to do little practice ones first. These are not real gels. I'm going to kind of take it out of my petri dish here. Kind of just a rubberized version of it, but the reason I like these is because it's more rigid. It's got the little openings here that we're going to be going through and putting our loading dye into, and we're going to do a method called submarine loading, and that's kind of already loaded right here, which means that we're going to fill it with water or buffer, and we're going to load inside of that. You're going to notice there's a penny on it. That's just kind of so that my little gel doesn't float up as it's inside that buffer. So I'm doing this one It's on a piece of yellow paper with a black line over it. And the reason I put a black line underneath of it is so that it's easier to actually see those openings rather than if it was just on the tabletop plane itself. So it's just a little easier to see when we have that. Sorry, I'm just kind of adjusting for you guys. Hopefully you get a little bit better view. And see if I can expand that out a little bit. So you can see all along that black line, we have 1, 2, 3, 4, 5, 6 openings that we want to fill. They actually are dug in. That's what we're making with the combs on the other side. So I'm going to go through. I'm going to uncap my loading die. It will stain your hands if you get it on you. I'm going to load my 8 microliters. I'm going to take off the lid, and there is actually water in there, as you guys can tell. And this wells and my gel are underneath the water. So what I want to do is I kind of want to just go on an angle. I want to stick my tip slightly into the opening. So if I have my finger on the plunger, as soon as I put it in, my finger is probably going to push it and it's going to start releasing. And I don't really want to do that. So I have it close to the plunger. I'm going to stick the tip barely in that little opening. And I'm going to go to first push. Very slow. It's a slow process. And I'm going to go all the way down to second push. Some of it will come out, but as you guys can see, that liquid is now contained within that little opening there, and it's going to kind of stay there. And we want to go through, and we're going to go through and do all of our solution, or all of our wells this way with the same solution. So I would go to the second one, go to the second hole opening, stick that tip slightly in, and I'm going to go to first push. And I want to go all the way down to second push. And I'm going to keep going through and doing it with all of them. So I'll try one more time for you. What I don't want to do, and what I see a lot of you guys do, is that you kind of stab and you want to go from the top and you want to go like this and go right through. The problem with that is this tip is very sharp. So if I'm going through and I'm going straight up and down, I'm probably going to stab right through my gel. And if that's the case, all my liquid's going to go underneath of it. These, you really can't do that to because they're so rigid, but with that soft sugar solution that we have, it is definitely much, much easier to puncture a hole in it, which is why I usually will go on the side. Once again, it is a super slow process. That loading die worked a little better for us, and we'll just keep going all the way across. Here's my fourth one. It is a heavy solution. As long as we're over that opening, it's going to fall into the opening. Keep my thumb pressed in so I'm completely out, and then I'm going to let go of my thumb. If I let go of my thumb when I'm still in there, I'm going to suck right back up what's inside that well. My fifth one. Keep my thumb in, second push, pull out. Now I'm good to go. I switched to loading dies about the third hole in, which is why it's going to look a little different to you guys when I take it off this colored paper. It's a darker loading die for you. My last opening. There we go. Pipette goes back into the rack. So as I slide that away, you can see that's where I kind of switched loading die, but that die is stuck inside those wells. So it's not like it's on top or the bottom of that liquid there. It's actually contained within the wells. So I say that's kind of the easy one. And now we're going to kind of go through and do what you guys did for your gel electrophoresis, or we'll be doing for gel electrophoresis. So once again, here's our gel boxes. Get a little better view of it for you guys. And I say kind of stand over top of it when you do it. This is really easy to see, this end one. You can see all those little openings there. But I have these same openings. You can see them in the middle there, and you can see them on the end there. So you kind of line them up with the red, or those red lines, and they're much easier for you guys to see. So we're going to kind of do the same thing. This one's a little bit more difficult, I think, because the gel is so fragile. But we're going to take 8 microliters, and I'm going to start on one end. So I'm going to start all the way up here at the top. And the biggest thing is trying to find that opening there. And we're just going to go slightly in. It's a very slow process, and we're going to slowly release the liquid, and it's going to sink into the well that we have. Go to second push and then pull out, and you'll notice that our first well is now completely full of, this is just loading dye, but next week we'll be doing proteins as well as some other substances. So we'll go through, we'll do the one next to it. Now, so if a little bit gets into the buffer, you're okay. Not going to contaminate it that bad, but what we don't want is to be way high up in the buffer and then start releasing it. And I'll give you an example of that in a moment. And what happens, instead of landing in the hole that we want it to be in, it kind of drifts over a little bit and it fills the second and the third hole. Well, here it's just loading dye, but if I'm doing things like proteins, I'm going to kind of cross-contaminate what I'm looking at. So what I mean like that, if I'm not quite in the hole yet and I just think I'm close by, I go, oh, let me just do it here. So it kind of just ciphers all over the place. And now it's filling that second hole or the third hole, and it's filling the fourth hole. So we make sure that tip is just barely into that little opening or well, and start to fill that. So I want to show you guys what happens when you stab the gel. So I'm going to go to a middle well here. Okay, so we're going to go to one of the openings in the middle well. And so if we were going through, and like I said, a lot of people like to go straight in on it. If I go straight in on it, I can't really see, and what happens is I start moving the gel and I go in, I go, oh, let me fill it. And you start moving the gel a little bit, and you don't want to do that. But what happens is you start pushing and you're like, nothing's coming out of the tip. It's because you clog the tip up with the agarose, and eventually it's like a little volcano that will release. And so what happens there is now my dye or proteins or DNA is stuck on the bottom of the gel, and that's not good. We want them in the wells like what we see here. So they're going to go through and kind of practice all of these. Next week when you do your first gel lab, you are only going to have mid-load wells, so the ones in the middle here, and we're going to go through and kind of load them full of all of our substances. It's called mysterious moving molecules. For that particular activity, we will go through and change tips for each one because we don't want to contaminate our samples. So as long as we're close enough, we're going to get all of our sample within there. So that's going to be their practice. They're going to be filling all of them. It's more or less just getting comfortable with it, and once they're comfortable with it, it's a good skill to have. They'll be doing it tons. Hopefully you guys will come back and we'll be doing it a tons there too, because we have ethidium bromide gels that we're going to do and some other DNA labs that deal with gel electrophoresis. So this is kind of hardened now. I want to show you what happens when I take it apart. I pull my little wells or my little dams on each end, and my gel in here is hardened. So I'm going to pull my little combs out, pull them out nice and easy because they are making the holes within my gel for me. Pull this last one. And now what I have on my little platform is this gel that you guys see here. So I can actually make these in advance. I can actually slide them off, and we'll be doing this when you guys do your gel labs because we'll be taking them out, put the little platform back, and now you are left with this very fragile gel that I can put into a baggie of some buffer to keep it nice and moist, and when I'm ready for them, I can stick them right back into our little gel boxes that you guys will be utilizing. Okay, so that's your practice loading of gels. Next lab you guys will see, it will talk about gel electrophoresis itself, so you'll see notes on it, as well as the first protein or the first gel lab that you have before we get to our protein lab.